science that comes out. So I hope you find this entertaining. At the end, I'll include my contact information if you have any questions. And so I live a bit of a double life for that reason, with both the science and the horror. That's my film scoring studio where I compose music for films, and I work at Sana Biotechnology. And I use viruses to cure cancer. Our drug got approved by the FDA in 2021, the last one we were working on. And then hopefully the next drugs will be approved soon. So we hear this term a lot, this virus is mutated in movies, and it's one of the main causes for zombie outbreaks, and happens all the time. And really common plot. And one of the problems with viruses is they're often misunderstood, but they're a great plot element for these films. So today I'll tell you a little bit about viruses, but also a bunch of other stuff. So as far as the most common plot elements, viruses are one of them, but you also hear about reanimation as a common plot element for zombies. You hear about parasites, another common element, and my favorite is when there's no room left in hell. This is my favorite reason for why zombies happen. So let's talk about viruses a little bit. And I mentioned we use viruses to cure cancer. And folks, I'm so sorry, I feel like I'm reaching out. I'm always in the way, out of the way, so I thank you for your patience. I should have brought my clicker, but I didn't. Thank you. This is how it works. This is our approved FDA product. We bring the cancer patients in, they are apheresed. So, and what that means is their blood comes out, and in a machine, part of their blood goes back in, but part of the blood cells are kept in the lab. Those are the white cells that are the immune cells, specifically the T cells. Mm-hmm. So we take those T cells, and then we engineer viruses to give them special genes. So that's a virus going in, a special gene going in with the virus. That special gene is expressed, and then we grow these cells in the lab. That gene allows these cells to recognize and kill cancer. We call the patient back 2 weeks later, 3 weeks later. So now they have a bag of cells that are infused into them through their vein. And it's their own cells, but it's been genetically modified with a virus. And they've been taught to now recognize and kill the cancer. So my team and I specialize in this part. There's about 12 of us in my team. We're molecular biologists, virologists, to make sure all this happens. And the drug is called Breyanzi. Lysocaptogene marlucil is the WHO generic name. So all drugs have a WHO generic name. And we started working on it about 2014, and it got approved in 2021. So now, what we're doing is, that process is complicated. It takes 2 weeks. So my team and I are now working on, can we just modify the virus and just directly give the virus? So yes, we are gonna cause the next zombie outbreak. And the way we do this is modify the outside of the virus and go from there. So let's talk a little bit about what exactly a virus is. We see a lot of images like this, and they're really cool. These are artist renditions, but viruses do really look like this. Not in color, they're never in color because they're too small to reflect visible light. Mm-hmm. But the one on the left is a bacteriophage that infects bacteria, and it does look like this, and it literally sits like a space shuttle, and then it squats onto the cell, and through the center, it's able to inject its DNA into the cell. Fascinating. The one on the right is just a boring flu virus, but it kills tens of thousands of people every year, more some years. And we were always worried that the flu virus would be the big pandemic. So the CDC and everyone have been following the flu very closely every year, but it was a coronavirus for the last one. In fact, some of the big ones in the last 20 years have been a coronavirus, with SARS 20 years ago, and then MERS about 10 years ago, and then now this one, SARS-CoV-2. We think of viruses as pathogens all the time. That's a favorite painting of mine on the left by Gabriel von Max. I actually saw the real painting at Henry Art Gallery here in Seattle a few years ago. It's beautiful, but it's about, and it's called The Anatomist, about our helplessness in the face of disease, and even physicians' helplessness in the face of disease. The two gentlemen on the right described the term contagious fluid to describe virus in the very beginning, and they identified viruses in tobacco mosaic leaves. And when you look at Wikipedia for the most deadly communicable diseases, all the yellow ones are viruses. Not all of them are viruses on the list. I know it's very hard to see, so I'll help you through some of them. So on the very top is prion diseases. Prion disease is the so-called mad cow disease. It's not a virus. Before the cause was identified, people thought it could be a virus, but it was spreading through simply eating meat, specifically certain meat from certain countries. And then it was identified as prions. Prions are, the person's got a Nobel Prize when they identified it. Prions are just protein. We all have proteins. All life is made of proteins. And the fascinating thing about prions is it's a protein that is slightly misfolded. So imagine a Lego piece, but you just kind of move a few pieces around, still within the rules of physics and chemistry, of course. And if that Lego piece, or if that protein comes next to the regular Lego piece or protein, it causes the shift in that, and the shift in the next one, and so on. And this triggers a whole chain reaction. It's still the same protein, but the structure, the folding has changed, and that causes plaques. After a certain accumulation, the plaques end up in the brain. So in the food cycle, it's simply acquired through ingestion. And we ingest it, and the reason the whole prion outbreak occurred is through recycling of the same meat. And how does that happen? That happens through cannibalism. So mad cow-like diseases were known in humans in Papua New Guinea. It's called Kuru disease, K-U-R-U. And Kuru disease was simply because these foldings would occur just randomly in the population, but they wouldn't affect anyone except that person. But if somebody ate that person, either through cannibalism or other means, then that protein continues in the food cycle. So the same thing happened with the cows, because the food that the cows were getting were partly dead cows as well, just included in the thing. So for this reason, most people from the United Kingdom aren't able to donate blood today, because there's the fear that some of them who are still alive, who lived in the UK during then and ate that meat, might still carry this. And in our research, we actually use cow serum to grow cells. This is very common. It's been used for decades in science. But we can only source cow serum from Australia and New Zealand because the cows in those continents have not been affected. They've been geographically separated from the UK. So that's the prions, and it's really cool stuff. And the list goes on, but when you hear viruses, rabies is the deadliest virus to humans. It's almost 100% fatal if you don't get treatment. Luckily, you can get treatment. And there is one person who's technically survived rabies even after the symptoms. They were put under a coma. And the reason people die is you get encephalitis in the brain and then you die from paralysis. They were put into an induced coma, completely experimental. The doctors just said, all right, we're putting her in a coma, this young child. She survived, and this was in Milwaukee. The treatment is called, for that reason, the Milwaukee Protocol. It was applied to many other rabies cases around the world, but it didn't really work with everyone. So we don't really know if that induced coma actually was a secret, or if she survived for who knows what. But it is so rare that the fatality, it tops the list of viruses. And it's my favorite virus. I had to get a rabies shot as a kid because I lived in Turkey, and we were playing with a cat, and the cat died. It turned out it died of rabies. So all of us neighborhood kids went to the hospital, and we had at the time, not anymore, an actual rabies hospital, so dedicated to rabies cases. So I remember sitting in the lobby. It's like waiting at a dentist where everyone has a story. And I remember hearing this couple next to me. I was a child, so I was terrified and fascinated. Cujo had just come out. I'd seen it before. There was this other Turkish film that was about kids in a village getting bit by rabies, but then there was no hospital nearby, so they're having to trek for 3 days over mountains and rivers to get to the rabies hospital. The clock is ticking, and then kids one by one undergo rabies. So it's terrifying. It was a terrifying film. That was a Turkish film. And these parents brought their baby in because some rat apparently gnawed the baby's face, and they were there getting a rabies shot. So And now I love horror films, probably because of that experience, and I'm a virologist, probably because of that experience. But this book is terrific. I recommend it. It talks about the cultural history, werewolves, and so on were probably all inspired by actual rabies cases, so I highly recommend the book. But what a virus is, in its heart, is nucleic acid, which is either DNA or RNA that's transmitted and that replicates, and it requires the host cell to do this, so it's either us or other mammals or bacteria or plants. And they come in many shapes and sizes. Ebola looks like a string. The flu looks circular. The cool thing about flu is instead of just one piece of genome, it has 8 pieces of genome. So if you have, say, a farm where cows, ducks, and humans live together, you can often have a bird flu, a pig flu, and a human flu happen to infect the same host. And then those 8 segments can recombine by chance, and all of a sudden you pop out a brand new flu that nobody has ever seen before. So that's why flu has always been the concern of pandemic. And if you ever hear the letters H5N1, H1 in the news, that refers to, on the surface there are 2 proteins, hemagglutinin and neuraminidase, and H stands for that one, the H, and then N stands for the N, and through the recombination, you get different types that pop out, sort of like a deck of cards getting reshuffled. And our immunity could be naive to some of those, hence the concern that all of a sudden something could spread. That's why you hear in the news about chicken farms where they'll kill millions of chickens just to make sure something doesn't come out if they identify a new thing like this. Not just humans, but also plants, bacteria get infected. So TMV, tobacco mosaic virus, is the first one that was identified, the first virus identified also in plants. We always thought viruses were small, and they are. They're about 100 nanometers, and on the bottom right is a scale. So that moon-like thing on the bottom is a human cell. On the right is E. coli bacteria that we have in our gut. And then on that top left dot in that same image is rhinovirus, which causes cold. We all get it, cold accidents. And then next to it is HIV. So those 2 gray dots up on the top are HIV and rhinovirus. So we always thought viruses were in that size, those 2 dots above, until these 3 blue guys were discovered. So they were discovered in ice from the Arctic, and they were discovered in these permafrost samples that were dated to be about 30,000 years old. And the virus, it wasn't just floating around on its own, it was frozen in ice inside amoebas. Amoebas, if you ever look in pond water under a microscope during your zoology, biology classes, they're those really cool single-celled organisms, and they're infected by viruses as well. So these viruses infect amoebas, and they turn out to be giant. They are almost as big as bacteria on the right, and we had never seen viruses that big until these papers started coming out. And you don't even have to go to the Arctic to see them. We actually have them probably on top of this hotel in the air conditioning unit with the water sitting there. Because why? Because amoebas are in that water as well. So these viruses are probably in those amoebas, and lo and behold, a few years later in France, they also saw the same virus in those amoebas in water containers, basically on top of roofs. It doesn't harm humans, but it still is fascinating that you can find all these cool life forms frozen in ice, and that, of course, clearly is an inspiration for many, many films, not just The Thing. Then there is this concept of, well, what if let's say you take a virus, but it has a shell, it has some of these cool things, it requires a host to grow, but then there are these life forms that don't even need those shells. They are literally naked chunks of RNA molecules that don't need any shell of any kind. Under an electron microscope, they look like that, just strings of RNA. And they are part of our life form as well, and they cause disease in plants. That's where they were identified. Potato Stimble tuber viroid is the name of this one, and they're called viroids. and they cause disease in potato plants, as shown in the diagram. One healthy, one diseased plant. But viruses are around everywhere. They don't just infect humans, they infect practically every form of life. And the most abundant life form on the planet, a single drop of ocean water has billions of viruses. That pie chart is a single drop of what an ocean water would look like in terms of numbers. But because viruses are very small, of course, it turns out that we always thought other forms would be greater, but they're the greatest. And so I think of viruses as transmission of genetic information. And when you think about it, so are we. So those of us who have children, and if their children have children, genetically we're propagating. Those of us who don't have children, that's basically dead end of their genetics, right? So same idea, same with the viruses, they propagate. They don't require the complexities that we require, like Tinder and all that. They just get by in a much simpler way. So therefore, more successful, arguably, from an evolutionary standpoint. When you look at the phylogenetic tree of life, you see viruses practically everywhere. This is a very classic tree of life that was designated in the 60s, 70s. There are three forms: the eukaryotes, which is where we belong with animals; archaea are these form of microscopic organisms that live in extreme environments, often say Yellowstone National Park and such, and they're such a different life form that they get their own branch, and also genetically they're distinct that they get their own branch. And then bacteria. So all these forms of life are infected by viruses, and the infection of viruses don't just serve the virus, so the virus is winning by spreading its genes, yes, but also it serves the host. In fact, viruses that do serve the host tend to win better than viruses that do nothing. What I mean by that is, say, if any of those organisms gain an ability thanks to a viral gene that's now in it, that ability can give a selective advantage and cause better and bigger and greater things. And also, viruses hop between species sometimes, and we see that throughout the tree of life. So it actually has clearly helped speed evolution in terms of giving mutational advantages to all these species. We already have mutations every day in our body, and nothing comes out of it except in very, very rare cases we get cancer, or in very, very rare cases we get a benefit. And if that benefit gives us a survival advantage, then our kids have that, and then their kids have that, and then so on. Now you have people who look like us. Yeah. So we think of pathogenesis, but really the true success of a virus is those that leave an imprint throughout evolution. And if you think about it, a successful virus may not even have to replicate anymore. It can be part of a very dominant species as just a genetic bit of information, because technically that genetic information is spreading with that host as well. And we see many advantages of this, so I'll tell you a story about this in just a second. The image on the right was sort of the first understanding of this story. It's an image of an embryo of a baboon. And when they look under the electron microscope, they saw these virus-like particles shown in those two arrows and then zoomed in on the bottom. They didn't really know what they were. Why are there these virus-like particles in a baboon embryo? Is it an infection? Is it something else? So I'll tell you what that is in a second. When the human genome was completed and the first draft came out about 20 years ago, we found out that the human genes are only 2% of our entire DNA. And the rest of our DNA was called junk DNA at the time. They didn't really know what it did. With computational methods advancing, we can now ask, hey, what does this look like if you scan it against the current nature genomes that we have in the database? What does this from Semi's genome look like if you scan it? And by doing this iterative exercise, we now know that half of our genome, the whole left side of that pie, are all viral genes in our genome. So if you take, say, any of those sequences in my DNA, and if you blast it, it's called blasting, against the database of nature genomes, you get all these viral hits. And you do this over and over, and it turns out half of our entire genome is nothing but that. And when all this became quite obvious, it was quite fascinating that more of us is a virus, frankly, than really human. So when you talk about human genetics or genetic diseases, we're only talking about that little purple slice, that's it. Yes, there are some genetic diseases that are caused by the rest of the pie, but those are rare. But the actual genes themselves really come down to that 2%. So, and lovely titles like this, right? Now, how does this happen? This happens because some viruses actually do integrate into their host genome as part of their life cycle. Retroviruses are a great example of this. HIV, it belongs to retrovirus, the virus that causes AIDS. And it enters the cell. HIV on its own has an RNA genome. And once inside the cell, it undergoes reverse transcription into DNA. And this actually was so important of a finding that it won the Nobel Prize, because before that, all scientists thought life was DNA, DNA becomes RNA, and then RNA becomes protein. And this is sort of the dogma of molecular biology. These viruses taught us it doesn't have to be that way. It can go the other way. So once they make DNA, they enter the nucleus of our cell, that blue area, and in the nucleus, which is where our genome is, they integrate into the genome. And they don't have to do anything else. They can stay there for decades. That's why in, for example, HIV patients, you will not see any AIDS, say, happening for years sometimes, where that has already inserted and nothing is really happening. But then at some point, that latency phase switches, and then they start producing more virus. And then that spreads, of course, to other humans. But the cool thing about this is we can use this for an advantage. So we use this in gene therapy. And I use HIV. So we have HIV, but HIV has been modified so it doesn't cause disease anymore. And it only causes that dead end there. So it doesn't replicate, but it's the dead end of insertion. And we use that, for example, in that cancer cure I mentioned. And we deliver that special gene to recognize and kill cancer, just like that. Now imagine if that cell was your sperm cell or your egg cell. What would happen then? And then imagine you had kids after that. Well, this diagram explains kind of what would happen. So HIV, COVID, transmitted horizontally from person to person. But if your germline gets infected, and that's shown by a big red circle in that crotch there, if you see it, and what that means is your sperm or egg cell has that sequence integrated in the genome. So your children are made up of you and your partner's DNA, and that virus DNA now as well. So your children will have that virus sequence in their DNA in every cell of the body. So all of our cells have the identical DNA. It's just that some parts are turned on, some parts are turned off. That makes it a liver cell versus a heart cell and so on. But all of the cells will have that DNA. And then their children will have all that virus DNA and so on. So this is how we believe today we ended up with half of our genome full of virus fossils over millions and millions of years. And not only humans, but our relatives in the non-human primate family. So you see this in the baboons, the chimpanzees, and monkeys. So all of our relatives have these as well. So clearly, this has been happening through our common ancestors leading up to today. Now, they're sitting there, but what do they do? So I showed you this black and white image already, the baboon plus image under a microscope with these virus-like particles. That was in 1975. We didn't know what it did, but now we do. So some of these virus sequences that I mentioned actually give us an evolutionary advantage. They allowed us to be mammals. They allowed us to give birth through the placenta. And here's how that works. When we were all in our mother's womb, the placenta feeds us, nourishes us, important things happen. Many complicated layers. One of those layers, abbreviated SYN, syncytial trophoblast, is formed on the bottom right as shown by fusion of cells. Many, many, many cells coming together, creating a sheet of multiple cells, and they're multinucleated because all the nucleus are still there, so these multinucleated sheets because of many cells. Our muscles are the same too, they're multinucleated sheets. And it turns out the virus, one of the viruses in our genomes actually allows that to happen. So it's in our genome. It's called HERV-W, H-E-R-V-W, Human Endogenous Retrovirus W. Most of the virus sequence is dead. It doesn't really encode anything, but the part that makes the envelope of the virus, the outside portion of the envelope of the virus, is still active, and especially active during pregnancy, allowing the formation of this layer by helping the cells fuse. And then later, it was discovered in muscles as well. So all of our muscle cells are fusing and forming that multinucleated muscle sheet thanks to this virus that's in our genome that probably integrated in our genome 20 plus million years ago, because we see the same virus in our relatives, not just monkeys, non-human primates, but also cows as well, and a lot of other placental mammals as well. So clearly it happened in a common ancestor, and we all still carry it. It probably gave us the evolutionary advantage of being able to have birth through the placenta. So this, of course, created a lot of enthusiasm, and one of my favorite science writers, his name is Carl Zimmer. He writes a lot of books, but during COVID, he started writing in the New York Times. He basically said in Discover magazine, if not for a virus, none of us would ever be born. So those sequences in our genome, as our genome replicates, those sequences replicate as well. And we have these elements that are virus-like. They're called transposons, and these elements in our genome replicate by cut and paste or copy and paste mechanisms. On the right is a cut and paste where it pops out and then it's not there anymore, but it pops into another site. On the bottom, it replicates itself and then it pops in its sister into the nearby area. So this happens in our genome all the time. In fact, every new birth seems to have about 6 to 10 of these new things happening. So the human genome is constantly evolving with every birth for that reason as well. And this whole idea of the evolution of the human genome to do certain things, I thought was very nicely modeled in this film, Crimes of the Future. Have you folks seen it? Some of you have seen it, David Cronenberg film. I didn't like the film that much per se, but the concept was terrific. So let me tell you, so that was an example in humans of how these viruses do things to help us thrive and to help us evolve. Let me give you another example, which is in parasitic wasps. Parasitic wasps, they find a caterpillar and they lay their larvae in the caterpillar, hence the name parasitic. A few weeks later, those larvae pop out, but interestingly, the caterpillar starts taking care of the larva to the point where it doesn't feed itself, and the larvas thrive, but the caterpillar dies. And the larva then form a new parasite and so on. So it turns out this is actually happening thanks to, again, viruses. So these parasitic wasps also have viruses in their genomes, and they start doing important things in different parts of their life cycle. They're called endogenous polydnavirus, Brachyvirus, or nudibvirus. They look like this under an electron microscope. This is that zoomed in, it looks like this sort of hexagonal shape. They are delivered to the caterpillar with the larvae, and they suppress the caterpillar immune system so it doesn't reject the larvae. And it also seems to alter their behavior, so it allows the caterpillar to treat the larvae like its own, and so on. So a fascinating example of what these do. And another example is, so this actor Mickey Rourke and then that bacteria virus on the right have something in common, and that is Botox. If you've had Botox done, or if you know anybody who has Botox, then you can thank viruses. So Botox is a toxin that's produced by a bacteria named Clostridium botulinum. That's the paper on the bottom. That toxin, however, is produced by the bacteria. So this causes food poisoning. The very famous, like if you buy a can from a supermarket, if it looks all swollen, you shouldn't eat from the can, because probably the bacteria is in there and it produced gas. So it probably has the toxin in there, and it's fatal. It will cause paralysis and death. But micro amounts cause paralysis for, say, cosmetic reasons or other medicinal reasons, and that's what Botox is. just microdoses of that same lethal toxin. The gene that produces the toxin comes from a virus. So these viruses are called bacteriophages that entered these bacteria. Again, who knows at what point in their evolution, but it seems to be conferring an advantage to them, not by killing humans, but probably by killing other prey of bacteria for the advantage of the bacteria. So viruses have, my friends, shaped the evolution of life, and they've been all part of making us who we are today. So for the rest of my talk, so I talked about viruses, but we still want to talk a little bit about how we use them. So gene therapy is how I use them. I gave you an example of how I use them for cancer by delivering a gene. But you can cure other diseases, such as sickle cell disease is a genetic hereditary disease where the red blood cells look abnormal. Therefore, they can't carry enough oxygen. This is due to a mutated gene. You can deliver the corrected gene with a virus so that these people can now produce healthy red blood cells. And that's happening. And the human genome, so every blue strand here is each of our chromosomes, but the human genome is flooded with elements that we now know cause genetic disease, that are hereditary. This list is only some of them. It doesn't include all of them. But using viruses, we can try to fix a lot of these because of the whole integration cycle that I showed you. This is, on the bottom right, is the very first human that was genetically modified to cure a disease. The way it was done is their cells were taken out, and their stem cells in the lab were modified with a virus. The stem cells were grown in the lab, the modified stem cells, and then they were infused back to the patient a few days, weeks later. This patient had ADA SCID, which is adenosine deaminase deficiency, and SCID stands for severe combined immunodeficiency. So these are the bubble boys. If you remember the John Travolta film with the bubble boy, so the slightest human touch will kill these children because bacteria and viruses on our skin, which are part of our normal flora, are deadly to them because they're born with no immune system due to this genetic disease. On the bottom right is when you have carrier mom, carrier dad, and then the probability of 1 out of 4 children could now have both of these recessive genes inherited, therefore now have the phenotype. Thankfully today with genetic testing, we can reduce the chances of this happening both by educating parents, but also possibly knowing that an expected child might have the disease, therefore trying to help the child with advanced genetic technologies. Now she's cured, she's alive today, that's what she looks like. There was another patient that was 9 years old, treated the same time, and I love this photo because she drew this. And in the lab, in the hospital room, looks like there's a door, bed. She's getting infused, so that bag holds the stem cells that were modified. This photo is from 1990, and if you see, there's a hospital TV, and it reads Willow. So she was probably watching this while receiving the very, very first gene therapy in the world. So I already mentioned at the beginning of our talk that our therapy was approved for cancer, and it's for cancer, but it's the same idea that you genetically modify these cells outside of the body and then give them. I just came back from ASGCT, which is the Society for Gene and Cell Therapy. I was invited to give 2 talks there, and then also we presented a poster, and there were 8,000 scientists from all over the world, and the field is advancing so fast, it's remarkable. So you're hearing terms like CRISPR, and now today gene therapy, and so on and so forth. Have you heard of Crippen virus? So Crippen virus, you might remember from the film I Am Legend. It's a genetically modified measles virus to cure cancer, but it mutated, and that was the plot point for I Am Legend, which by the way is a remake of Omega Man, which by the way is a remake of The Last Man on Earth. And it's not a real virus, it was a fictional virus in the film, named after the scientist's last name, Crippen virus. But I love the plot because it's exactly what we're doing today, using genetically modified viruses to cure cancer, but in the film it went wrong, right? So it's a great plot point again. In the last minutes, let's talk a little bit about COVID, just so I can help all of you get all of us level set on what exactly happened. And here we are, practically most of us maskless. Masks still work, but life goes on. And I still wear a mask actually when I travel, especially. It's a coronavirus. Coronaviruses are called corona because under the microscope they look like the sun solar flares or the crown, hence the naming and so on. They have these spike proteins on the outside. So those of you who got a vaccine, it's against the spike protein. It's meant to trigger antibodies in your body and cell responses in your body against that spike protein. It has a genome, that squiggly line is an RNA genome. And there are lots of coronaviruses. We are infected by a lot of them, actually. Sometimes when you say you have the flu, you probably don't have the flu. You probably have a coronavirus or a rhinovirus or an adenovirus, which are the common cold viruses every year. But so all those that aren't in red are basically the common colds that we get every year that are still coronaviruses. We get them every year. They come from animals. So from animals, they get acquired by humans. You don't have to be in contact with the animal because you just probably got it from another human. But then SARS happened 20 years ago, and that was a big coronavirus problem. And for the first time, at least within modern day, people thought, oh shit, we gotta watch out for coronaviruses. And a lot of papers were published 20 years ago saying, at the end of the paper, saying, oh, therefore it's likely a future pandemic could occur because of coronaviruses. We know it came from bats. It came from bats to civet cats and raccoon dogs, which are these unique species, not related to cats or dogs, but called such. And they were being sold at meat markets. And they're illegal, their trade is illegal, but it was still happening in these wild meat markets. And clearly humans and those animals coming together in close contact. And they're being kept in cages for a very long time. So that contact is just a matter of time before the virus jumps to the human, just through respiratory droplets. Or sometimes their feces so the feces will dry and they'll become dust, and then humans inhale that dust, say cleaning the cage or sweeping the floor. So that's how it happens. And then we had MERS, which is from bats to camels to humans. That's why MERS is called Middle Eastern Respiratory Syndrome. It started in Saudi Arabia because of where camels and humans tend to cohabitat. And then 3 years ago now, SARS-CoV-2. This image isn't up to date. It shows a pangolin because for a while it was thought that it was from bats to pangolins to humans, but now we know that it's most likely from raccoon dogs again. And I'm happy to talk about that after this talk if you have any questions. But this is called zoonosis. And a lot of diseases have happened this way, from same with HIV. We talked about HIV a little earlier, to coronaviruses, to the flu. I gave the examples of how a pig flu and then say a duck flu, a bird flu can come together and cause recombinations and such. And as I mentioned, so just recently in May, through genetic testing and swabbing of this meat market where supposedly the origin of cases came from, they very much now have evidence that the outbreak, the virus, the humans, and then these raccoon dogs were all there at the same time at the same place. So there's still the missing link, the 100% evidence that yes, this is the cause, but this is the closest we've ever been, because no other theory. I mean, I've been doing science communication for 3 years, and a number of times people come to me saying it came from a lab, or it's caused by 5G networks, or it's caused by Bill Gates, or whatever. I've heard this so many times, and of course, it would be, all of those would be true if there's proof of it, but there simply has been no proof other than this is the closest evidence we actually have of what the likely cause may have been. So we'll see. If they actually capture an animal with a SARS-CoV-2-like sequence, obviously that would be definitive, but the markets were all cleaned up before any testing could be done, unfortunately. The coronavirus family, let me introduce them. They are, imagine this like grandparents and kids and so on. So they belong to this order, Nidoviralis, and there are these several families, Arterioviridae, Coronaviridae, Mesonaviridae, Ronaviridae, and so on. And if you keep checking out all these genera, SARS-CoV-2 belongs to this bottom genera under beta coronaviruses. So they're part of the beta coronavirus family. I'm sharing this only just to show you that there's tons of this stuff. And an outbreak is always imminent for that reason. It's just a matter of time before another bat comes in contact with another animal, comes in contact with us, and then three of them happened in the last 20 years, right? So the phylogeny, if you look closer to beta coronaviruses, are in blue. It's not very visible, but all those coronaviruses I showed you that cause human disease are mostly bundled under the blue beta coronavirus family. And the disease was deadly, unfortunately, to quite a few people. I forgot the latest numbers, but 6 million worldwide, 1 million in the US or something. I forgot the total death tolls, and very sad. Maybe some of you may have also been affected with families and so on, but the actual death is not by the virus. The virus isn't killing you. It doesn't secrete anything to kill you. The virus is just replicating, just like I showed examples of. It's just doing its thing. We're having children, they're having children, same idea. It's our immune system that's killing us, and here's what I mean by that. Our immunity evolved to be at a very fine balance. If you have too much of it, you have autoimmune disease like Crohn's disease, skin diseases, lupus, and so on. If you have too little of it, you have infections, you have cancer. So it has to perform right at this balance. And that balance moves and sways all the time with your life, of what you've been exposed to, but also with your parents, meaning your genes. And we basically, our body evolved to attack coronavirus when it sees it. Your body is already primed to attack coronaviruses because it's seen other coronaviruses already in your lifetime. So when it sees a new one, it just attacks. Now, unfortunately, due to a bunch of combinations, that attack tends to be more severe with some coronaviruses like this one. That severity leads to your lungs getting filled up with all the immunity and cytokines that are produced by the immune cells. And then that now causes damage to your healthy tissue. The damage to healthy tissue is part of your immunity. The idea is kill the neighboring cells so the virus doesn't have neighboring cells to move onto. But in this case, the speed of it, the severity of it, unfortunately causes these tissue degradation in your lungs, therefore lack of oxygen, therefore death, and so on. And not just oxygen, but many, many other problems, like the whole so-called long COVID disease that seems to now be a thing. The coronavirus life cycle looks like this. It enters the cell. It replicates in the cytoplasm. So unlike the other viruses I showed you, it doesn't enter the nucleus. The HIV example I gave you enters the nucleus, and that's why it's so good for gene therapy. Coronavirus doesn't. It stays in the cytoplasm, and then it pops more of it out. So these, my friends, are viruses, and we always hear about viruses mutating, but mutations are very normal. If you see headlines about mutations, just know that they're part of life. It happens all the time. And it's a way of evolution sampling errors. So trying a few things out, and sometimes those few things hit, and there's an advantage, and then we evolve. And it happened with coronaviruses as well. So through websites like this, you can track a lot of pathogens. This is an example of coronavirus evolution over time. So that's a tree again, the idea of grandparents and daughters of coronavirus, but now left to right instead of top down. And every dot is a sequence across the planet, as shown in the map. And those branches are now happening because of the evolution of the coronavirus over time. This is normal. Also on the bottom with the colors, you can see some of the new colors starting to take over and the old colors not around anymore. That's just the evolution of the virus. Those that have a selective advantage thrive, those that don't die off. The mutations that do help the virus the most are those that allow it to, say, attach better, and so on. If you zoom into what's happening at the surface of the virus in the cell, the proteins are graphed out like this. These are three-dimensional structures, and they literally do form these ribbons, and this is just the way the computer displays it. But in those interactions between, on the top, the reds are virus, in the middle, the green is the human receptor it binds to. You have these protein-protein interactions, and when one of them changes, it can now bind better. And on the bottom, you can take this whole thing and open it up as a string, and on the bottom in that string now you can see, okay, it's happening here, a change, it's happening here. That makes sense because it's right at the attachment site. So those are viruses, my friends. Let's talk a little bit about my other favorite categories, and I'll make sure you have enough time to get to your next session, but reanimation. And So I got my PhD at the Fred Hutchinson Cancer Center working on viruses and HIV, but this professor is also there, and he figured out how to reanimate dead tissue and dead animals. And the story is, it's not a green liquid, it's not a Jeffrey Combs green needle, but it's basically a type of gas. And it turns out the secret is the following: when we die, tissue breakdown happens immediately and brain damage happens immediately. So the idea is, if you can delay that, you can buy time. And you can buy time and theoretically, with medicine and in a hospital, bring the person back with minimal decay of the brain, which is the most important thing. So he figured out how to do that. And then there were all these US Army generals thumping through the Fred Hutch Cancer Research Center where I was at, because he now got a grant from the military to do this research. Why the military? Because the idea that in the battlefield you could inject these people with that green liquid and now buy time to get them to the hospital to either help them recover, or if they're dead, hopefully bring them back. And he showed this happen in all sorts of animals. He would literally kill them and then they would come back. And the secret is the residual amount of oxygen left in our system when we die and we stop breathing apparently is really toxic. Through means of all the oxidative species and so on and so forth. They're very complicated, but the trick is apparently to even deplete that. So that 0.1 whatever percent of oxygen left in your body, you got to deplete that. And luckily there are other gases that you can add that will competitively replace oxygen in your red blood cells. And that gas turned out to be the secret. The clue from this came from, there was a popcorn factory somewhere, and these people, there was a factory accident, and these people died, but then they came back to life. And they came back to life because the gases that were released in the popcorn because of the artificial flavoring was basically had the same chemical. So these people were inhaling this all the time, every day. It's not harmful to you. You gotta make sure you have enough oxygen, of course, but when they died, it was to their benefit. So that was one example. And then there were examples of people in caves who were exposed to similar gases in caves, died and come back. And of course, temperature and other things are important. Like you may have heard of stories of people who fell into frozen lakes and then they came back. So that's a real thing. Parasites are a real thing. We talked a little bit about parasitic wasps, but there are these parasites in nature that are spread through bird poop into fish, and then normally fish that swim in the deep, it alters their behavior and they come back to the surface to be eaten again by birds, hence the cycle continues. Wow. And of course this, my friends, which some of you may have seen, this is The Last of Us, the HBO show that's really popular based on the video game. There's a YouTube video, that's the scan of it if you want it, but on BBC, this is from a few years ago, but this is a fungus, and it causes in ants and many other insects, it's doing the same thing, just like we are, it's trying to reproduce. And ants are going about, but then they start doing weird things, and they start climbing up above for no reason. And then these stalks start protruding from their bodies. These are all the spores. And you see this not just in ants, but all other sort of insects. There's a moth on the bottom and all these sprouting bodies. So that inspired the video game, I suppose, and the movie. And if you look at the science of it, that's what the fungus is called, and the actual full name. And then in the life cycle of the ant, it helps with the life cycle and reproduction of the life cycle and so on. So there are a lot of these guys, and you can basically look at the phylogenetic tree. So that's the same species, and there's a ton of them, and they all do different things with different life forms. So it's a real thing, and it's a fascinating, of course, show today. So my recommendation for you, if you're interested in this stuff for further reading, Parasite Rex and Planet of Viruses are both by Carl Zimmer, that same author I mentioned, who I really like to follow. He's on New York Times. And Rabid is a great book about the history of rabies and how it influenced us. And then film-wise, Contagion is probably the best accurate viral pandemic movie. Still, of course, exaggerated, but the terms they use are real. And Antiviral is by Brandon Cronenberg, David Cronenberg's son, where people buy celebrity viruses to infect themselves because that's what celebrities want to do. So, thank you, my friends. I'm going to end my talk there, but I'll stick around for questions. And I appreciate you listening this morning. Thank you so much.